So hello everyone, my name is Ankit Sharma and our problem statement ID is SIH25042. So this is our proposed solution. In this we have used the dataset provided by organizers which included blast data basis for reference. We extracted data, linked taxonomy and prepared a pipeline using 18S and COI marker genes that cover protists and eukaryotes. So our proposed solution is Equinox. Our solution is Equinox, an AI-driven pipeline designed to classify deep sea eDNA without over relying on incomplete databases. So here's how it works. So step one is basically treat DNA as language, split each DNA sequence into three mer tokens, just like breaking down a sentence into words. Now in step two, here you can see that we have pre-trained BERT on DNA using masked language modeling. We mask 15% tokens and train the model to prepare them. Using next sentence prediction, we shuffle 50% of sequences so the model learns context. In step number three, this is step number three. We fine tune for taxonomy. We add classification heads that predict superkingdom, phylum, and genes simultaneously. And in the final step, that is step four, we have inference flow that tokenize DNA, encode with BERT, summarizes heads output probabilities. This means instead of matching to a database, Equinox learns DNA grammar and classifies even unknown organisms. So we have some additional feature also. Now here in the PPT, you can see this is system architecture. So to align with the PS requirements and add impact, Equinox doesn't just classify sequences, it also annotates sequences with taxonomic labels across multiple ranks, estimates relative abundance of taxa to reveal biodiversity distribution, identifies novel organisms by detecting sequences with no database, matched by but consistent DNA grammar patterns. So finally, why Equinox is better? So Equinox overcomes three major challenges. First is database dependency. It classifies sequences without relying on exact matches. Novel taxa detection works even when organisms are not in databases. Now, third one that is most important, that is speed and scalability. Optimized AI reduces computational time significantly. In short, Equinox enables real-time biodiversity insight that traditional pipelines cannot. Now, let us move to the website part. So, this is our website. This is our team name, Team Trioton. So Ocean eDNA. First, here you have to upload DNA file. So I select this DNA file. This is sample deep sea DNA in FASTQ format. I'll open this file, and once the sample is validating it, I'll start the AI analysis. So it will take some time. So result generation is done, and we have identified 347 taxa other than 28 novel taxa, and this is most important thing here. The analysis time is reduced by 95%. That is 18 minutes. We have done it this analysis in just 18 minutes, and quality score is 36.8. And this is biodiversity matrix. What is Simpson index? What is species richness, evenness? We can see, and here also Protista, Animalia, fungi, and unknown species. What has been detected from our FASTQ format set, and we can see that these are 10 most abundant taxa here. The list of 10 most abundant taxa, and even with the percentage mentioned. Now we have rare taxa discovery. You can see that these graphical representation also makes it visually very pleasing. So here we can see AI insight, deep sea adaptation, ecological roles, conservation priority, and what depth we have analyzed at 4,200 meter. What was the temperature? It was 2.1 degrees Celsius. Salinity, and further these are PDF reports, CSV data, JSON results, FASTA sequences, and here you can see that the AI pipeline advantage that we have done this analysis in just 18 minutes. In traditional, we have to take around 12 hours for this, so it is 40x faster processing. Finally, these are all other related insight, and this is also that if you want to share some results, so we can share it directly from this link. If I start generate link here, so I can share, if anybody wants this analysis, I can share directly this link and it will be shared to him, this website insight. So after this is our AI research section. Here you can visualize the pipeline, how our pipeline works. So in the last section, you can see this is visualizing the clusters, understanding abundance, identifying dominant organisms. And finally, we have our map section also. In the map section, once you click any particular water body, you can find the taxonomy of the ocean. So once the data set will be completed, it will be fully functional. Now we come to this part. This will tell about the accuracy of our model, how accurate our model is. So here you can see that what kind of data set we have taken, what were the tools, and how much similarity we have got for each of the data set that we have taken. So thank you everyone.